Okay, we are just going to let everybody trickle in for a few minutes and then we will get started. I'm going to go ahead and share my screen. Okay, I'm going to just wait another minute or so and then we will get started. Okay, hello everyone and welcome to the Asian Carp Canada webinar series. My name is Leanna Hranowitz and I'm the Aquatic Program Coordinator at the Invasive Species Center and I will be your moderator for today's webinar. Before we begin, I'd like to acknowledge that the Invasive Species Center honors the long history of the First Nation, Inuit, and Métis Nation people on the lands now known as Canada and strives to show respect to their ancestors, communities, and to them individually. The Invasive Species Center respectfully acknowledges that our head office is located on the traditional lands of the Anishinaabe, the Batchewana, and Garden River First Nation, as well as the longtime settlement of the Métis people in the Robinson-Huron Treaty area. The Invasive Species Center is a non-for-profit organization that mobilizes action against invasive species that harm the environment, economy, and society. We've got lots of great invasive species resources on our website, including species profiles, best management practices, and more, so check us out at invasivespeciescenter.ca. We also have a newsletter you can sign up for to hear more about upcoming webinars such as this one. Asian Carp Canada is a program under the Invasive Species Center. On our website, you can find species profiles to all 4 invasive fish, risk assessments, past webinars, current research, and much more. Before we get started with today's webinar, there are a couple of things I'd like to mention. There will be time for questions at the end of the webinar, so if you have a question at any time, please type it into the Q&A box and I'll read it out to our presenter after the webinar. If you're having technical difficulties at any time, please type them in the chat box or respond to the email found in your registration and we'll try to resolve it as quickly as we can. We also have enabled closed captioning, so if you'd like to follow along that way, you can turn on the closed captioning button with your taskbar. Lastly, there will be a brief survey following the webinar, so if you could please take some time to fill it out, we would greatly appreciate it. So today's webinar is titled Early Detection Surveillance of Grass Carp, Captures and Response Efforts, and will be presented by Trisiah Tugade. So Trisiah is an aquatic biologist with the Asian Carp Program at Fisheries and Oceans Canada, located in the department's Ontario and Prairie Region. Trisiah has a master's degree in environmental science from the University of Toronto, where she has worked on research projects concerning non-physical barriers to aquatic invasive species. She started at DFO as a Fisheries Protection Biologist with the Fish and Fish Habitat Protection Program, and now works in the Aquatic Invasive Species Unit. As the aquatic biologist for DFO's Asian Carp Program, Trisiah conducts fisheries field sampling, laboratory work, and data analysis and report writing for early detection surveillance efforts in Canadian waters of the Great Lakes Basin. So with that, I will pass it over to Trisiah and thank you for joining us. And I will stop sharing my screen.
Just gonna share mine. All right, hopefully that looks okay.
Looks good.
Okay, so thanks, Leanna. As introduced, my name is Trisiah. I work with DFO. I'm here with the Asian Carp Program, here to talk about early detection surveillance of grass carp, captures and response efforts, and I'll be focusing more on the 2023 field season and what's been happening in the past year. So it wouldn't be a talk from the Asian Carp Program without honorable mention from the other Asian carp species, not just grass carp. So along with grass carp, we have big head carp, silver carp, black carp. And these 4 species, as you can see, get these enormous sizes as displayed on your screen. Black carp get the biggest, weighing over 200 pounds, getting well over 1 meter in length. I think some people are familiar with silver carp, where those are the ones in the viral videos jumping out of the water when they're perturbed by a boat motor and posing significant risks to boaters. And each of these Asian carp species eat different things, so they pose different pressures on the food web. Big head eat zooplankton, silver carp eat phytoplankton, and black carp eat mollusks, while grass carp eat aquatic vegetation. So you'll notice that common carp is not on this screen. It's not part of the Asian carp species, and we often get misidentification and misreports from citizens and anglers about common carp. I'll show a video later on detailing the different ID features of grass carp and how you can distinguish it from common carp. But a main feature is looking at that short dorsal fin on each of these Asian carp species and comparing it to a really long dorsal fin, the fin on the back of a common carp. You'll also notice that none of these Asian carp species have those fleshy whiskers or barbels, while common carp do. So to give context about why we care so much and why our Asian Carp program was established and why we put so much effort and allocation into surveilling our Great Lakes for these species is because these aquatic invasive species are able to outcompete our native fishes for resources. So each of those Asian carp species are voracious eaters. They're eating machines, consuming 20 to 40% of their body weight per day. And so consuming, all the aquatic vegetation, phytoplankton, zooplankton, mussels, snails, freshwater shrimp. These are all critical resources for our own native fish to survive. On top of being eating machines, they're also breeding machines, and that they're such prolific breeders. They can produce up to 1 million eggs a day, or sorry, not a day, in a spawning season, and in turn produce thousands of offspring. So these two behaviors, their spawning and their feeding behaviors, you can understand why they're able to dominate habitats should they invade Canadian waters of the Great Lakes. In the states where they are established populations and where they're in much higher numbers, a lot of the time they outnumber their native fish 10 to 1 and can make up 80% of the biomass of the fish population. So you can see how that would significantly and negatively impact those ecosystems. So as promised, I'll be focusing more on grass carp for this talk. And yes, all 4 of those Asian carp species are pose such a huge threat and an imminent threat to the Canadian waters of the Great Lakes should they invade. But grass carp are our most immediate threat, the ones that we focus a lot more in our program, because there are reproducing populations of grass carp on the western basin of Lake Erie on the US side. So they're knocking right on the front door of Canadian waters, and I don't think they respect international borders too much like us. So that begs the question of how grass carp got here. Well, they were purposely introduced in the 60s and 70s from China and Russia, and as I said, they are such eating machines. So they were purposely introduced in aquaculture ponds as a method to control aquatic vegetation. And due to flooding events, they escaped. And when they escaped, they were able to make their way to the Mississippi River river system, where they were able to spread through its tributaries and make its way up northward. And now they're up north as the Illinois River and its tributaries, so right on the front door of the Great Lakes. And this is a great slide to showcase some more fun facts about grass carp, and that you don't need many fish to establish a population. 20 fish could overtake the Great Lakes, and that comes from the fact that they're such prolific breeders. They can also grow up to 4.5 feet long or 1.4 meters, and eating 40% of its body weight in aquatic vegetation a day lends itself to gaining 2 pounds a month. So this slide shows the year 1 and year 2 age class, and you can see just how quickly they're able to grow. And should they, arrive and establish here, they can quickly outgrow the mouths of any natural predators and find themselves without any natural predators in our Great Lakes. And this brings us to the Asian Carp Program and why our program was established. So due to the threat of these Asian carp species, due to the threat of grass carp, we were established in 2012. And this was a multidisciplinary program. And evidently our goal was to protect the integrity of the Great Lakes by preventing the arrival, establishment, and spread of the Asian carp species. So since the inception of our program in 2012, we work really diligently with our US partners to protect Great Lakes from these highly invasive freshwater fishes. We also have a very significant and successful collaboration with Ontario's Ministry of Natural Resources. And it's important to note that this joint responsibility of aquatic invasive species in the Great Lakes is a joint one between the federal and the provincial governments. So our program sits on 4 critical pillars: prevention, early warning, response, and management. And all due to the sake of time and also the focus of this presentation, I'll be focusing more on early warning and response, which is all about our Asian carp field program and where our captures come from and how we respond essentially. But of course, prevention is just as important. That's the purpose of why I'm doing this webinar today. And it's all about preventing the threat from entering Canadian waters through public outreach, education, and awareness. And we do this with the help of our partners, of course. So Ontario Federation of Angler and Hunters, or OFAH, of course, the Invasive Species Center, ISC, and the Toronto Region Conservation Authority, TRCA. The prevention pillar also encompasses the science and research and risk assessments, and we'll see that how that it helps us inform us on the other pillars as well. The prevention pillar is so important because of this graph neatly demonstrates that. So we see on the y-axis the prevention or management costs and time on the x-axis. And so as you can see, we're at the bottom of this curve right now where we don't have established populations of grass carp in our waters. We're trying to prevent that. And right now the cost is at its lowest. But should we wait? Should we delay action? Should we wait until they're established? The cost for managing this species would just be way more costly than the time, resources, and money that we're putting into now. A really great example of this is the sea lamprey program. So sea lamprey are already established in the Great Lakes, and our sea lamprey program, we put a lot of time, money, and resources into that program now that they're established just to manage that population. So all the more reason why it's critical, why we all stay vigilant, why we have our early detection program, because we want to keep costs lower and prevent ourselves from progressing along this curve. We're going to move to the early warning pillar now, and this is all about our Asian carp field program. We really want to detect that threat early, the grass carp, through surveillance alongside partners and efforts of key organizations. And again, the key organizations are MNRF and TRCA. And of course, our objective is to find any of these Asian carps, grass carp, more specifically, that may be present and remove them safely from Canadian waters. Our 2 goals, of course, is the early detection of grass carp, but also while we're out there doing sampling, surveillance, and all these tributaries of the Great Lakes, we're doing a full fish community assessment. So that means we're processing every fish we catch, and this serves as a pre-impact baseline so should these Asian carps or grass carp invade, then we have all this historical data on the native fish community present, and we can see how that could trend over time with these negative impacts. As you can imagine, looking for grass carp across the Great Lakes Basin is like looking for a needle in a haystack. We need to expertly deploy our resources and time and crews into select locations where we're most likely to find them. So luckily, science, research, and risk assessments has helped us inform us of these early detection surveillance sites. So these 37 sites listed on the map have been deemed as suitable spawning habitat or habitat for Asian carps or grass carp to hang out in. So on a year-to-year basis from May to November, we're out there with our early detection field program, surveilling these sites using a variety of different traditional gear types And we are sampling the river mouths, the wetlands, the nearshore areas, and slightly up the river in these different tributaries. This map on the left just shows that in the risk assessment of the 79 tributaries that were studied in the Great Lakes, 57 were deemed to have suitable spawning conditions for Asian carps. So how are we doing our surveillance and sampling? Well, there's those traditional gear types I discussed. So bag seines, trap nets, hoop nets, and fyke nets. And all you need to know is that these traditional gear types are deployed depending on the habitat present. So is there a lot of vegetation? Is it more shallow? Is it deeper waters with more flow? And are we looking for smaller bodied fish or larger bodied fish? Boat electrofishing is a favorite in our program. So we do a lot of boat electrofishing and sometimes in tandem with gill and trammel nets. So we'll encircle an area with a gill or trammel net and we'll boat electrofish inside and outside and process all the fish that are either captured in the net or captured by the boat electrofishing. We also do larval sampling. So we look for egg and larval fish in our program. And this is done with light trapped and bongo nets. So on top of looking for the individual grass carp species, adults or adult-sized grass carp, we also look for their eggs and larval grass carp. And we haven't found any larval or egg from grass carp to date in Canadian waters, but it is something in our arsenal and something that we deploy on a year-to-year basis. So this is just going to give you an idea of kind of the breadth of our sampling on a year-to-year basis. So these are statistics from our 2023 field season that just passed. So we did 161 hoop nets, 12 bag seines, mini fyke nets, 267 sites. We also did trammel and gill nets, 77 sites and 21 sites respectively, and trap nets, 148 sites. As I said, boat electrofishing is a favorite, so we did 488 sites, and that totaled to just over 113 hours of boat electrofishing. For our larval sampling, we set 161 light traps, and those are more geared towards capturing larval grass carp, and the bongo nets can also capture larval grass carp, but more so are looking for eggs. So 71 sites. This is just a map showing where we did our larval sampling last year in 2023. So we did 8 high-risk tributaries that were deemed to have very suitable spawning habitat locations. And we did 232 sites each, and that totaled 186 samples containing egg and or larval fish. As you can imagine, it'll be hard to distinguish what larval grass carp would look like from other species when they're that tiny. So we send that for genetic testing, and the results on that are still pending. So this table just gives you a sense of our sampling since the inception of our program. And so 2020 and 2021 were impacted by COVID operationally, but since then we've been able to bounce back and do over 1,000 sites. So 2023, we were able to cover 1,174 sites in our early detection surveillance, covering 38 water bodies and 84 species. You'll note that buffalo and common carp are counted individually, and this is because they're considered surrogate species for us. So these are great indicator species in that they're also large-bodied fish, similar to grass carp, that occupy similar habitat. We're not capturing grass carp in really high numbers. In fact, we're capturing them often in, single-digit numbers. So in order to for us to infer, are we looking in the right habitats, and are our gear being effective? We like to tally the amount of buffalo and common carp. Grass carp there is that last row. And it's important to note that these numbers are just the numbers that were collected by our early detection surveillance program. So this does not include the commercial fishermen, the recreational anglers, or citizen reports over the year. And I'll show that in the next slide. But all grass carp captured by our program were captured by trammel or gill nets. So this slide shows how many grass carp have been captured since the inception of our program in 2012. And again, 32 since 2012 really shows how little these grass carp are in numbers in our Canadian waters, and why it's important to stay vigilant, and why we put so much effort into looking for these grass carp. Every little bit counts. So drawing your attention now to the 2023 numbers here. So 3 were captured just last year, and I can get into a bit more detail in these next few slides. So the first one that was captured in our 2023 field season was in the Grand River, and this was captured by our own program, the Early Detection Field Program. And this was captured June 20th of last year. It was almost a meter in length, and it weighed 8.3 kilograms, and it was determined to be a sterile male. Here it is just noted on the map in the Grand River where it was captured. The next one was the Bay of Quinte. This was captured on July 3rd of last year. This one was a whopping 21 kilograms, and unfortunately it was found to be a fertile female. So it was gushing with eggs. So this was in the Bay of Quinte, right here in this purple location. And lastly, for our grass carp captures in 2023, we have the Niagara River. Oh, sorry, I just want to note that the Bay of Quinte one was captured by a commercial fisherman actually, and reported to us, where we took it back to our lab, did analysis. I'll get into that in a bit. The Niagara River one was captured by a recreational angler, and it was captured on August 16th, and it weighed 12.6 kilograms. It was determined to be sterile, and the sex is to be determined. And here it is. Here's a site of capture. So now that we have gotten into showing the captures over the years and in the 2023 field season, we'll get into what exactly happens when a grass carp is captured and how we respond as a program. So this video gives you a little overview of the response actions and our response pillar before I go into much more detail in a few other slides.
Trisha, I don't know if we're able to hear the volume on it.
Oh, you can't. How do I? Can't hear the volume on the. Thank you to my helpful colleague, Mon, in troubleshooting some IT. Hopefully this works now. I'll restart it from the beginning.
The Asian Carp Lab is really the command center for responding to detections of Asian carps in the Great Lakes. and all of the information we can collect on these fish is done in this lab here. My name is Julia Combe. I'm an aquatic biologist with Fisheries and Oceans Canada's Asian Carp Program. My main role is conducting fertility testing on the specimens and then doing a full dissection to get as much information from every individual as we can to help better understand where these fish are coming from. Asian carp pose a huge threat to our Great Lakes ecosystems. They can reproduce really quickly, multiple times per year. They produce a huge number of eggs at any given time. And the very first thing that happens when one is captured is it comes back to the lab and we do fertility testing. And if it is fertile, we're going to follow up with operations on the water. You want to detect them early and remove them quickly to prevent those species from having the chance to reproduce and establish a population. I feel a lot of pride knowing that I'm part of a team that's protecting not only the Great Lakes, but the inland waters beyond as well.
Okay, great. So that video gave us a really good overview of response actions, but I'll be going into a lot more detail about what exactly happens when a grass carp is captured. So part of our response pillar is when that grass carp is captured, it gets transported back to our lab here in Burlington. And as a biologist, I'm responsible for figuring out whether this fish is fertile or sterile. And we run blood samples or some of the eyeball fluid in this really fancy machine called a flow cytometer, and it spits out some results and tells us whether it is fertile or sterile. And depending on this result helps us inform program management decisions. So as you can imagine, a sterile fish, one that's not able to reproduce, doesn't pose as much of a high risk as a fertile one. A sterile one can impose negative consequences in its ecosystem, right? It can eat a lot of aquatic vegetation, dispel a lot of waste, and decrease water quality. But that's over the course of its lifetime. Whereas a fertile fish is able to possibly lead to an established population, one that's reproducing. And instead of just imposing negative consequences over its lifetime, it'll impose consequences over many generations. So depending on this result, we will enact strike teams to exhaustively sample the area, which I will get into a bit more detail. So on top of doing the fertility testing, we also do full dissections of this grass carp. So in the lab, we do measurements, we get weight, we take photos, and we also cut it open to remove the gonads to do gonad histology. We do some aging of the structures like the scales and the vertebrae or the dorsal fin to give us an idea of course, the age of the grass carp. And there are these really tiny bones inside of the skull of the grass carp, pictured here. They're so tiny compared to this enormous grass carp, but they tell us so much about the fish. So we can send those otoliths, one of them for oxygen isotope analysis, and that can help us inform us of where this fish originated. These otolith bones come in pairs, so then the other one can be sent for microchemistry. And we can match this microchemistry to other microchemical signatures within the Great Lakes to figure out where this fish was hanging out throughout its lifetime and before we captured it. We can also do fin clips on the fins, and that can be sent for genetic testing, and also take the stomach contents to examine the grass carp for its diet. Finally, the muscle plug can be taken, and we could do contaminant analysis. So as you can see, just from one fish, we can capture so much information, why it's so important that every fish that's captured or reported is transported back to our lab so we can do all of this analysis, and really feeds into our program management decisions. So as I said, this response when a grass carp is captured, we when it's captured, we actually activate this system called the Incident Command System, or ICS. And it's just a really fancy term for organizational structure that helps us streamline decisions, communications, and respond effectively to a grass carp capture. And it also helps us to maintain organization as we coordinate with our partners on the water. So once the fertility of the fish is determined, we have our incident commander that will tell us whether or not we need to exhaustively sample the area. So if it's a fertile fish, we will send out strike teams to sample the area and remove any grass carp that are present within the vicinity. And once we feel like the threat is eliminated, we'll stop. If it's a sterile fish, then we actually don't do on-the-water operations, but it is important to note that we still respond with our notification protocol. So all our partners are notified, and each of our partners have their specific target audiences with a great reach. So they're able to spread the word on our behalf, notify everyone about the capture location, and to keep everyone on the lookout and keep their eyes peeled. So when I say response operations and deploying strike teams, this will just give you a sense of what that looks like. So from 2015 to 2020, here's a few of those response operations and what that kind of looked like. So in the Toronto Islands in September of 2015, 3 fertile grass carp were captured. One of them, 2 of them were captured from the response itself. One of them was the one that initiated the response. And in this response, 3 different agencies responded. So DFO, MNRF, and TRCA. And this resulted in 8 crews or 8 boats of having up to 20 staff on site per day. And this was over the course of 4 days. We also have the Lake Gibson response in June of 2016. 10 grass carp were pulled out over 10 days using 3 crews between DFO and MNRF. Lastly, the Jordan Harbor response, the most recent one, besides the 2023 responses, which I will get into shortly. Just one grass carp, deployed or a fertile female, was pulled, and this triggered a response in Jordan Harbor over 6 days with 3 crews from DFO, and no additional grass carp were captured from that response. So now I'm going to get into the 2023 responses. So if you don't remember from their 2023 captures, of those 3 grass carp, one of them was found to be fertile. So the fertile female was found in the Bay of Quinte. And the Bay of Quinte is over here in this Belleville area, zoomed in over here. And so when we found that fertile female, we enacted our ICS and we deployed strike teams within this area. So here's where it was captured, just showing another historical capture that happened in 2015. And this was a sterile one, but this fertile one was the one that triggered our response actions. So in July of 2023, we sent 3 boat electrofishing crews, 2 from DFO, 1 was from MNRF. And while we were there, we also collected eDNA from the Trent River, which is in Trenton. And while we were doing our response operation, the sampling was targeted in spawning and shallow wetland habitats throughout the Bay of Quinte, not just within the site of capture. Sampling went on for 5 days, and no additional grass carp were captured from this response operation. So this just gives a spread of the different sampling sites during our response operations. So here's the Trent River in Trenton, the Moira River here, and then we have a whole host of other broader broader sampling sites where we stuck to the most suitable preferred habitat for grass carp. And it's important to note that grass carp that was captured by the commercial fishermen have these commercially sized trap nets, so they're able to access the deeper waters, which is how the grass carp was captured. Our traditional gear types are more geared towards the shallower depth waters, so we stuck to more of the shallower habitats during this response, and that suitable preferred habitat for grass carp spawning and for grass carp to hang out in. So just some statistics of what that effort looked like. So total effort, we did just over 33 hours of boat electrofishing, 100 minutes of trammel netting, and from the eDNA, there were 3 sites that were collected for eDNA from the first dam upstream to the mouth of the river. And so there was also this August 2023 response. So MNRF performs a regular routine eDNA sampling throughout the Great Lakes, but in response to the captured fish of the Bay of Quinte fish in July, there was some heightened eDNA sampling, and that we suspected that maybe there would be some positive hits. So there was 2 positive hits that were collected from this eDNA sampling in Muscogee Bay, just noted on this map. And it's important to note this was MNRF's eDNA sampling. This was not from the eDNA that was collected while we were doing the response in July. And here's just a spread of those eDNA sites where it was collected, and the 2 red ones are the positive hits. So our response, although because we did not have the fish in hand, that response was not at the same level of urgency as a captured fish in hand. But still, we still sent out a strike team, so consisted of 3 boat electrofishing crews between DFO, MNRF, and Quinte Conservation. Trap nets were also deployed along the shoreline, and the efforts were focused within the Muscogee Bay, so around those positive hits. And 5 days of sampling had occurred. The purple are the trap nets and the green are the electrofishing sites. So there were 42 trap net sites. It just doesn't look represented on this map because as soon as they were pulled, they were replaced again. So there's actually multiple trap net sites within each dot. And for boat electrofishing, just over 14 hours were completed for this response, and 42 trap nets, as I said, totaling almost 100 hours. No additional grass carp were captured in this response. So this all brings us to why we're doing this webinar, why it's important. Yes, I'm here to provide updates on our 2023 field season and captures, but it's always important to inform everyone here about how you can help and how you can get involved. So our early detection work is very important, but I think everyone plays a role, and tips from the public are essential. Not every grass carp captured were captured by our program, but they were captured by, citizens and recreational anglers. So if you see or catch a grass carp, please report it. First step, obviously, do not release it into the water. Take a photo, try to confirm the identity, and we'll go through some grass carp ID features. Try to note the exact location as well. And then you want to report it once you've confirmed that it is indeed a grass carp. So you want to call Invading Species Hotline at 1-800-563-7711, or you can report it to edmaps.org/Ontario. Once you've reported it, you want to, again, make sure you don't release it. So you want to retain it. So you want to keep it in a cooler, head above ice if possible. And you don't want to mess with the head at all because, as I said, those otolith bones that are in there that tell us a lot about the fish, you want to prevent those from being lost. So wrap the head in a wet paper towel, keep it above ice. And because you've reported it, hang tight and someone will come to pick it up and transport it back to our lab. So this is going to go over some really quick ID features of the grass carp. And so you can see the eye is level with the mouth, has no barbels or those fleshy whiskers, a short dorsal fin, and has that large dark cross hatch scale pattern. And you can tell this apart from common carp because common carp have that long dorsal fin versus this short dorsal fin, and it has those barbels as well, whereas grass carp don't have those. And then just a reminder of that phone number, should you want to report, should you come into contact with a grass carp and report it, and also the email for invading species, info@invadingspecies.com. And I think this video is really great. I like to show it because it really drives home the point of those key features of grass carp. So I will play it.
Grass carp are often confused with common carp.
So this is a juvenile grass carp, approximately 30 centimeters in length, but they do reach up to 1.5 meters in length. And while this guy's under a pound in weight, they do reach up to 90 pounds. Grass carp has a terminal mouth, which means it ends right at the end of its head. Not facing down, not facing up, but just right off the end. This fish does not have any barbels at the side of the mouth. The scales have a darkening on the back of the scale, which actually creates kind of a cross-hatch pattern on the side of the fish. Now, when most people observe these fish, they're going to see them from above. And how we distinguish between the common carp and the grass carp is with the common carp, the dorsal fin would start approximately where my thumb is here and run back to here, where it's much shorter with the grass carp. So the grass carp has the short dorsal fin, common carp has a long one. And that's the easiest way to really tell them apart when they're in the water. Our team takes every report seriously and we act upon it. We have a chance to make a difference here. There's only a few fish out there, and we want to get them early and get them out of the system. So every set of eyes on the water really helps this effort.
Grass carp?
So that was a really great video going over the ID features. And before we end, I just want to throw this map up to please keep an eye out. So this map shows where historical captures have been and where grass carp have been reported to be seen. So whether you're around these areas, but also I think whether you're in, on, around the water, please keep an eye out. And these high alert areas are critical, but also across the Great Lakes Basin is important. So I'll end there and I'll throw my contact information up on the screen. If you want more information, please visit asiancarp.ca, but also feel free to reach out via email should you not get a chance to get your question answered or think of something later on. Thanks.
Thanks so much, Tricia. That was an awesome presentation. We do have a couple questions in the Q&A box, so I will read those out to you. And if anyone has any questions, to please pop them in that Q&A box and I'll read them out. So first question, what does gonad histology mean?
Oh, so we basically examine the eggs or the testes of the grass carp, and we're just looking for maturity of those structures to see, you know, are they reproductively like viable and how mature they are.
Great, thanks. This question you touched on a little bit, but maybe you can reinstate it. Have you considered eDNA sampling as an alternative to electrofishing and netting for early detection? It would also potentially capture larval stages and results can be produced in as quick as 10 days.
So yes, I feel like eDNA, everyone is very much excited about eDNA right now. And it's a great tool, but I think it's just that it's one of the tools in the tool belt, and I don't think it could, it's not exactly the entire tool belt. So it's a great support for it, but when we use eDNA, there's so many factors to consider in terms of, you know, the flow of the water, or how long the fish has been hanging out in the water, or size of the fish, other fish present. So eDNA is a great tool, yes, but I think because of the complexity of it, the traditional gear types are very much our primary focus in our early detection program. And yeah, so we'll continue to, support MNRF's eDNA project with eDNA efforts. But again, I think it's just one of the many tools that we use for early detection surveillance.
Great, thanks. Is lab testing only done for grass carp at the moment, or for all Asian carp species that are captured, including the other types?
Well, we haven't had a other Asian carp species transported in our lab for analysis. So at the moment, I can only speak to, the work that we do on grass carp in our lab. So yeah, sorry, that's as best of an answer I can give. But I'm sure, if other Asian carp species would be captured, we would perform the same sort of recording and tests in our lab.
Great. What is the origin of the Bay of Quinte fish that was captured? Was it different than the Lake Erie records? And are people still using grass carp for vegetation control in Ontario? A couple questions.
So it's actually illegal to possess grass carp in Canada. It's part of our regulations, so no. In the States, it's legal to stock in certain states, but they have to be sterile. In certain states, I can't give you specifics on which ones off the top of my head. And then the origin of the Bay of Quinte fish. So I've sent off the otoliths for testing, but unfortunately I don't have those results at the moment. They're still pending. And sorry, I think there was another portion of that question.
Yeah, it was related to the origin of the Bay of Quinte fish, if it was similar to that of like the
Okay.
records.
I see.
Okay, thanks. So this question's asking about the samples that were classified as sterile. Is there a chance that they would be just immature and not sterile?
I don't think so. Our methods with a flow cytometer I think are robust enough to indicate that it is indeed sterile and not just immature.
Okay, after a capture, if a grass carp's head does become frozen, does analysis of the head become impossible or just more challenging?
It just becomes more challenging, I think. So those otolith bones are really tiny, and we're just trying to make sure that they don't get dislodged should you tamper with the head. So it's possible to find it at times, but sometimes they do get lost in the head and then we lose the bone and we can't send those off for analysis.
Awesome, thanks. What would cause sterility in a fish?
Oh yeah, so it's a great question. When they were stocked in the States, they were stocked as sterile individuals. So they have a method for sterilizing grass carp by shocking the gonads, and it's not 100% effective method to sterilize a fish, hence why you have fertile fish as well. And so these sterile fish escape when there's flooding events, and hence why they're found in the wild.
Great. Have any other species of Asian carp been observed to leap out of the water like silver carp, or has it just been a behavior observed in silver carp?
I believe that's just silver carp.
Great. And I am not seeing any other questions, so I think we will wrap it up there. Thanks so much, Tricia, for joining us today for the webinar. This webinar has been recorded and it will be posted on www.asiancarp.ca. And just a reminder, you'll be sent out a survey after the webinar, so if you can please fill out this survey, that would be great. I'm also going to mention that we have an angler survey live on our website at the moment. So if you are an angler or a water user, if you can maybe take some time to fill out that survey, and you will actually be entered to win a $100 Bass Pro Shop gift card for entering the survey. So that's also on our website. And with that, we will conclude it there. And thanks again, Tricia, for joining us.
Thank you all for listening. I enjoyed it.